I'm going to talk today about muscle wasting, which is an enormous clinical problem in many different conditions, ranging from the age-related muscle wasting in the clinic, in the intensive care. Now with the new drugs for obesity loss and the accompanying muscle wasting. And I've been working on regulation of muscle contraction for almost 5 decades, presently at Karolinska Institute, but I've also been fortunate to work in multiple other universities in Europe and as well as in the US. So we had idea is actually to try to understand regulation of muscle contraction from the whole muscle down to the motor protein and motor protein modifications, and also develop methods and models where we can go between clinical studies and also in experimental animal models. And as you can see here, from the whole muscle to the motor unit to the muscle cell down to the motor protein level, motor proteins being post-translational modified by using mass spectrometry as well as X-ray diffraction experiments which we've done at the Spring 8 synchrotron in Osaka, Japan then. But also looking then of course at gene expression. And this is just showing one of the models we're using to look on regulation muscle contraction on the control conditions in human muscle cells. And we use exactly the same model when we look in experimental animal models. And also we have a model where we can extract the motor protein from single cells, and in this small in vitro motility assay, where we can then look at how fast myosin can propel actin, and then we have a molecular measure of maximum velocity or unloaded shortening. But also by adding actin binding proteins, we can also have a handle on the force generated by myosin without any secondary influences from structural proteins that we have at the single cell level. And more recently, I've also been working with Professor Gina using these quantum dots to also look at the myosin efficiency. And here we're not already at the moment we're not down to the single muscle cell level, but that's what we're aiming for at the moment. But I also like to tell you a clinical problem that we are very interested in, and this is the muscle wasting and complete muscle paralysis that we see in intensive care unit patients. To the left here you see a normal muscle cell from a patient under relaxing and under maximum activating conditions, and to the right you see from this patient. And this is the first patient we diagnosed with this in Scandinavia when I was on call Christmas '95. It was an elderly woman that had become quadriplegic in response to modern critical care. And I was there actually primarily to do electrophysiology, which indicated the motor axonopathy. But I also heard about this disease. We took a muscle biopsy, went back to the lab, pulled out single cells, and to the right you can see these cells under relaxing condition and also under maximum activating conditions. And these cells developed zero force. A normal cross-striation pattern was lost. And then we solubilized the cells and looked at the protein expression. And we could then see that the motor protein in the muscle cell was lost. There was no myosin in the muscle cells, while other proteins like thin filament proteins, actin, etc. were fully expressed. And this is just showing the same thing at the EM level. To the top left corner here you have the control biopsy, we have the C lines, and in the center you have the A band where we have the motor protein. And the other two slides you see from these patients when we see the C lines, but there's no A band in the center, and that's where we have the molecular motor protein myosin. So it was not so surprising the patient was completely paralyzed. And then try to understand mechanism. And first of all, maybe I should say, at that time, it was considered to be a rare condition of limited clinical significance. We can see these patients in any ICU. And after we found the first patient, just by looking around in different ICUs at the Karolinska Hospital, we found more than 10 patients. And nowadays we can go to any ICU and find these patients at different severe or milder forms then. But to try to understand mechanisms, we started with trying to use a porcine ICU model. And later on we moved to a rat model, which has the advantage that we can do long-term experiments, which is very important to try to understand mechanisms, but above all also when we test different intervention strategies. And what I would like to talk about a little bit more today is actually how this is also affecting the major inspiratory muscle, the diaphragm. Because almost 40% of the time intensive care units are mechanically ventilated. It's just weaning the patient off the ventilator. And some of these patients stay on the ventilator for weeks and months because the diaphragm is too weak for the patient to spontaneously breathe. And ICU costs are enormous. And these patients in the US has been estimated to cost $64 billion annually. And this was before the pandemic, so you can imagine what the costs were during the pandemic. So we then looked at how this intensive care unit condition in the rat model affected regulation of muscle contraction. And 10 days mechanical ventilation, the residual force generated by these cells was less than 15% of normal. So it's not so surprising that it takes a long time to wean these patients off the ventilator. It's also important to remember, of course, that mechanical ventilation is a life-saving intervention in intensive care unit patients. And typically what is the preferred mode of mechanical ventilation is what we call with assisted mechanical ventilation, where the central drive sort of assists the spontaneous breathing because it induces less lung injury. But instead of this, in the general ICU population, almost 30% of the patients develop this critical illness myopathy. But we have in 2 independent studies looked in neuro ICU patients who do not have this central drive, where we have to use controlled mechanical ventilation, where the ventilator is controlled ventilation completely. In both these studies, all patients develop this critical illness myopathy with preferential myosin loss. And also this, I think, is related to actually the lung injury induced by the mechanical ventilation. So then we started an intervention. We looked on bone marrow-derived mesenchymal stromal cells, which generate biologically active extracellular vesicles that has a restoring effect of protecting injured lung cells. So we did a study in this rat model, and what we could see by giving these extracellular vesicles, it has a very positive effect on the lung, and with a much less lung injury, which was also coupled to improvement of diaphragm muscle function. It reduces the diaphragm muscle cell wasting, but it also improved force generating capacity, which is the maximum force generated by a single diaphragm muscle cell normalized to muscle fiber cross-sectional area. So it improves both size and function. And doing multi-omics, genomics, proteomics, and metabolomics, we could also show restoring effects of this EV treatment in lung, the diaphragm, but also bronchoalveolar lavage, the fluid that comes out of the respiratory tract. And this is something we can also use for both for diagnostic but also for monitoring purposes. And at the moment, this is an intervention which we are aiming at translating to a clinical study as well. So thank you.